Antibiotic resistance is a growing health problem. For instance, the bacterium Streptococcus pneumoniae, pneumococcus, accounts for hundreds of thousands of deaths each year and is now multi-drug resistant in a sizable proportion of infections. We have found a new and practical way to counter the spread of antibiotics resistance by inhibiting the exchange and spread of resistance genes among bacteria, using low concentrations of non-toxic, FDA-approved molecules. How does this work? Bacteria become resistant by modifying their genes or by taking up resistance genes from fellow bacteria. Transformation of DNA can occur when a process called competence is activated. As soon as a bacterium gets hold of resistance genes, it will survive antibiotic therapy, and it lives on as a new, resistant strain. Some compounds can stimulate competence. These compounds include antibiotics. If, for some reason, these don't fully kill the bacteria, this helps them to acquire resistance to these same antibiotics. We reasoned, if you can stimulate competence, you might also be able to suppress it. So we set out to find molecules that do exactly that. To test for success, we used genetically modified pneumococci. As soon as competence is switched on, they start making luciferase, a light-producing enzyme. This way, they light up as soon as competence starts. After testing 1,286 commonly used bioactive compounds, we identified several compounds that reliably switched off the luciferase glow. This means that they suppress competence. When we administered such compounds, along with free-floating resistance genes, the bacteria were no longer able to take up these genes. That means that transformation was inhibited, as was the spread of antibiotics resistance. Also, in closer to clinic conditions, living on a culture of human epithelial cells, our competence blockers prevented pneumococci from becoming resistant even when resistance genes were present. Many of our new competence blockers are non-toxic and FDA approved. We propose to use them as an adjuvant to clinical antibiotics or vaccines to help prevent resistance from spreading and killing people.